Hello everyone. I hope you all are doing great. I, Pooja Verma, welcome you all in the third session of chapter 9, that is Biomolecules. Learning objectives of today's session are to help you learn about the nucleotides, which is the monomeric unit of nucleic acids, to help you familiarize with the structure of DNA, to help you learn about biocatalysts, their active sites and turnover number, to help you understand the metabolic basis for living and how enzymes enhance the rate of reaction, to develop scientific attitude, creativity, aesthetic sense and problem solving approach in you, to enable you to find the applications of these topics in your daily life. Nucleic acids are main information carrying molecules of the cell. Nucleic acid is an important class of macromolecule found in all cells and viruses. The name nucleic acid comes from the fact that they have acidic properties, much like the acids you know, and the nucleic part comes from the fact that they were first isolated from nucleus of cells by a Swiss scientist, Friedrich Mischer, in 1869. By directing the process of protein synthesis, they determine the characteristics of every living being. Nucleic acid is of two types. They are deoxyribonucleic acid or DNA and ribonucleic acid or RNA. DNA is the master blueprint for life. It encodes the information the cell needs to make proteins. It constitutes the genetic material in all living organisms except certain organisms such as retroviruses. Viruses which contain RNA as their genetic material are called retroviruses. RNA is also found in all living cells. It is of three types: messenger RNA or mRNA, ribosomal RNA or rRNA, and transfer RNA or tRNA. It plays an important role in protein synthesis. Nucleic acid molecule is made up of large number of nucleotide molecules joined into a linear and unbranched chain. Nucleic acids are thus polynucleotides. Nucleotides are formed by joining of nitrogenous base, sugar and phosphoric acid. Nitrogenous base can be either purine or pyrimidine. Purines are large, 9-membered double rings as you can see on the screen. It has nitrogen at first, third, 7th and 9th position. It is of two types, adenine and guanine. Pyrimidines are smaller, 6-membered single rings. A pyrimidine ring has nitrogen at first and third position as you can see on the screen. It is of three types, cytosine, uracil and thymine, commonly abbreviated as CUT. Nucleic acid contains either ribose or deoxyribose sugar. Ribose sugar is present in RNA. Its molecular formula is C5H10O5. Deoxyribose sugar is present in DNA. Ribose sugar differs from deoxyribose in having a hydroxyl group instead of hydrogen at second carbon. A combination of a nitrogen base with a pentose sugar is known as nucleoside. Observe the structure of adenosine shown on the screen. For nucleoside, we use the suffix sign if nitrogen base is of purine type and suffix -dine is used if the nitrogen base is of pyrimidine type. Nitrogen base is linked with ribose sugar through glycosidic bond. When nucleoside joins with phosphoric acid, they form nucleotides. For nucleotides, suffix -ilic acid is used. Nucleotides join with the help of phosphodiester bonds to form nucleic acids. DNA is the largest macromolecule in the organisms. It is associated with histone proteins and found inside the nucleus. A small quantity of DNA also occurs outside the nucleus in mitochondria and plastids organelles. Such DNA is called extracellular or organellar DNA. DNA is a long double chain of deoxyribonucleotides which consist of nitrogenous base, pentose sugar and phosphate. DNA contains adenine, thymine, cytosine and guanine nitrogen bases. Uracil is absent in DNA. Thus, there are 4 types of deoxyribonucleotides in DNA: adenosine monophosphate, that is AMP, guanosine monophosphate, GMP, cytidine monophosphate, CMP, and thymine monophosphate, TMP. Two nucleotides join with the help of a phosphodiester bonds to form a linear unbranched chain. J.D. Watson, an American biologist, and F.H.C. Crick, an English physicist, in 1953 suggested a model to explain DNA structure. They were awarded the 1962 Nobel Prize for this work. Let us try to understand the DNA structure. In each chain, the phosphate component carried by the carbon atom at 5' position of sugar in one nucleotide is joined by phosphodiester bond to the hydroxyl component of the carbon atom at 3' position of the sugar in the next nucleotide unit. These 3' to 5' phosphodiester bonds help in the formation of polynucleotides. The alternating sugar phosphate forms the backbone of the DNA helix. Can you tell me the name of the bond which joins the nitrogenous base with pentose deoxyribose sugar? Yes, it is the glycosidic bond. As you can see on the screen, a DNA molecule has 2 unbranched complementary strands which are coiled around a common axis like a spiral staircase with base pairs forming steps, while the backbone of the 2 strands, which is made up of sugar phosphate, forms railings. The backbone of a DNA is built up of alternate sugar and phosphate group. Phosphate group is connected to the 5' carbon of the sugar of its own nucleotide and 3' carbon of the sugar of the next nucleotide by phosphodiester bond. Water is eliminated during the formation of ester bond. At one end of the DNA strand, the last sugar has 5' carbon free, while at the other end 3' carbon of the first sugar is free. These are respectively called 5' and 3' ends. Its polarity is 5' to 3'. Polarity of the second chain will be 3' to 5'. Thus, the two chains of DNA molecule run in opposite or anti-parallel directions. It is similar to a two-lane road where the lines run parallel but they carry traffic in opposite directions. The two nucleotide chains of DNA molecules are not identical but complementary to each other with respect to base pairs. Base pair refers to two bases, one in each chain of the DNA molecule, joined with the help of hydrogen bonds. A base pair must consist of a purine and pyrimidine. A specific purine pairs with a specific pyrimidine because of a perfect match between hydrogen donor and hydrogen acceptor sides on the two bases. Adenine pairs with thymine with the help of two hydrogen bonds, while cytosine pairs with guanine with the help of 3 hydrogen bonds. The diameter of the DNA helix is 20 angstrom. Two purines or two pyrimidines cannot pair with each other. This is consistent with the fact that there is not enough space for two purines to fit within the helix. If two purines will try to form a base pair, it will cause overcrowding. In case of two pyrimidines, the distance between them will be more, thus they cannot form hydrogen bonds and join with each other. And so the two DNA chains are complementary to each other. The helix is right-handed, that is, the turns run clockwise looking along the helical axis. One complete turn of a DNA helix is 34 angstrom long, which contains 10 base pairs. So, the distance between two consecutive base pair is 3.4 angstrom. We will now come to the topic of enzymes. I hope you all know what are enzymes. In chapter 8, you had studied that lysosome contains various types of digestive enzymes. Enzymes are proteins that act as biological catalyst or biocatalyst. They increase the rate of a chemical reaction without itself being altered in the process. The molecules upon which enzymes act are called substrates, and the enzyme converts substrates into products. Most of the enzymes are made up of proteins except ribozymes. Ribozymes are special enzymes made up of RNA. They have a specific three-dimensional tertiary structure. The part of the enzyme where the substrate binds is called active site. An active site of an enzyme is a pocket into which the substrate fits. The active site consists of amino acid residue that forms temporary bonds with the substrate in a reaction. All the active sites occupy only 10 to 20% of the volume of an enzyme, but it is the most important part as it directly catalyzes the chemical reaction. Active site binds to a particular substrate and catalyzes a particular reaction. This makes enzyme highly specific in their action. This specificity is determined by the arrangement of amino acids within the active site and the structure of the substrate. While enzymes are biocatalysts, there are also inorganic catalysts. Such inorganic catalysts and enzymes exhibit similarity in some aspects. For example, both are required in small quantities. Both increase the rate of reaction. They do not undergo any change in their composition and quantity, thus they can be used many times. Inorganic catalysts and enzymes also differ in many ways. For example, inorganic catalysts work efficiently at high temperature and high pressure, whereas enzymes work efficiently at normal body temperature and normal pressure. Inorganic catalysts are not affected by poisons and are less efficient catalysts, whereas enzymes are inactivated by poisons and are very efficient catalysts. A large number of chemical reactions takes place in the living cell all the time. Without enzymes, these reactions will be so slow and will not be able to sustain life. Due to enzymes, various chemical reactions can occur at normal human body temperature and normal body pH at a fast rate. Most enzymes have a high turnover number. A turnover number of an enzyme refers to the number of substrate molecules acted upon by one molecule of enzyme per minute. Turnover number of carbonic anhydrase is 36 million. This enzyme is present in the RBC. It helps in the transport of carbon dioxide in the form of carbonic acid during respiration. As you can see on the screen, it helps in the joining of carbon dioxide and water to form carbonic acid. High turnover number of enzyme explain their remarkable effectiveness even though they occur in minute quantities in a cell. Higher the turnover number, the more efficient the enzyme is. In chapter 1, you have already learned that metabolism is a characteristic of living organisms. In a living cell, thousands of reactions are occurring at any given point of time. Metabolism refers to the sum total of all the chemical reactions occurring in an organism at any given point of time. All living organisms need energy to grow and reproduce, maintain their structure, and respond to their environments. Metabolism is a set of processes that make energy available for cellular processes. It includes catabolic as well as anabolic processes. Anabolism refers to the process in which small, simpler molecules join to form large, complex molecules. Catabolism is the reverse of anabolism, in which breaking of complex substances into simpler substances occurs. Respiration is a classical example of catabolism. During respiration, breakdown of glucose occurs in the presence of oxygen and energy is released, which is trapped in the form of ATP, that is adenosine triphosphate. ATP is the energy currency of the cell. Respiration provides energy for performing various activities. Without respiration, no living organism can survive. A metabolic pathway is a multi-step interlinked series of enzyme-catalyzed biochemical reactions in which a product of one reaction generally becomes a substrate for the next reaction. A metabolic pathway is a series of connected chemical reactions that feed one another. Glycolysis and citric acid cycle are the classical examples of metabolic pathways. There is a separate enzyme for every biochemical reaction. Enzyme catalyzed reactions are hundreds of times faster than an uncatalyzed chemical reaction. Thus, without enzymes, life would not be possible. As you can see on the screen, when barium hydroxide reacts with sulfuric acid, it forms barium sulfate and water molecules. How do we calculate the rate of a reaction? Rate of a reaction is a measure of how much reactant is consumed or how much product is produced in a given amount of time. It is determined by the amount of product formed per unit time, that is delta P divided by delta T. Substrate concentration, temperature and the presence of enzyme or catalyst are some of the main factors which affect the rate of a reaction. Rate of a chemical reaction doubles for every 10 degrees Celsius increase in temperature. Thousands of biomolecules occur in organisms at a characteristic concentration. The living system maintains this concentration of biomolecules because they are in a metabolic flux, always remaining in non-equilibrium steady state. You might have studied in chemistry that no work can be carried out in equilibrium state. Therefore, living organisms continuously receive an input of energy and try to prevent reaching equilibrium so that they can do work. Energy is obtained from metabolism. Hence, the living state and metabolism are synonymous. Without metabolism, there cannot be a living state. Let us now discuss how enzymes increase the rate of a chemical reaction. A graph is shown on the screen. On the x-axis, progress of reaction is shown and the y-axis represents the potential energy. Most of the chemical reactions do not start automatically because the reactant molecules must overcome an energy barrier to become reactive. Majority of the molecules of each substrate have average kinetic energy, some have higher, some have lower than the average energy. Under normal temperature, only the molecules which have high energy are capable of forming products. Therefore, the reaction takes place very slowly. One method to increase the rate of the reaction is to raise the temperature of the mixture by heating. This will increase the kinetic energy of the molecules and thus the rate of reaction. But this approach cannot be used by a living cell, as we have to maintain our body temperature constant at 37 degrees Celsius. So, how does the cell solve this problem? For substrate to be converted into products, substrates must reach transition state. In this state, substrate bonds are broken and new bonds are formed. The difference in average energy content of substrate from that of this transition state is called activation energy. Therefore, an external supply of energy is needed for the start of chemical reaction, which is the activation energy. To solve this challenge, cell uses enzymes which are biocatalysts. Such biocatalysts increase the rate of reaction by lowering the activation energy. Catalytic activity of the enzyme is due to specific regions on their surface called active sites. These small pockets are formed as the enzyme molecule folds upon itself to acquire three-dimensional structure. Active site contains different amino acids having positive or negative charges. The size, type and the nature of electrical charge, the amino acid R group decides which substrate can fit inside it. The charges inside the active site attack the bonds of the substrate molecules by various electrophilic and nucleophilic mechanisms. This develops strain in the bonds of the substrate molecules. This results in the formation of unstable intermediate structure called transition state. In this state, substrate bonds are broken and new bonds are formed, which helps in converting the substrate molecules into products. Once the activation energy decreases, the rate of the reaction automatically becomes fast. Let's have a quick recap. In today's session, we have discussed the structure of nucleotides and their joining through phosphodiester bond to form DNA. We have also learned about the structure and function of enzymes and also discussed the mechanism through which enzymes increases the rate of reaction. Before we conclude the session, here is an assignment for you. Draw a well-labeled diagram of DNA. Identify the type of bond present in the following: between two nucleotides, between nitrogen base and sugar of nucleosides. If enzyme X has a turnover number of 2000 per second, while enzyme Y has a turnover number of 6 lakhs per second, which enzyme will you consider a better enzyme and why? With that, we will conclude today's session. Hope you all enjoyed the session. Keep smiling, keep learning. Thank you.